Welcome. This is Dr. Ed Park from Recharge Biomedical, and I want to thank everybody for taking the time out of your day. I know it's a very, very busy time of year. The topic is, how does TA65 work? As a first principle, we should discuss what a telomere is. A telomere is the end body of our chromosomes, and it serves to protect. Uh, every time a cell divides, the telomeres must shorten. It's a mechanical thing by about 50 base pairs. So as the cells divide, they eventually get too short, and then that's a problem. The paperclip is a pretty good actual image because the end of the chromosome, as you can see here, folds back on itself just like a paperclip, and that's how the end is protected. If we zoom in a little more on the right here, we see that there's the red strand and the blue strand. The blue strand is what we call the 3' strand. That's the overhanging strand. And it always matches the red strand.
Running in circles, chasing tails, coming back as we are.
So if it's an A here, it's a T here. If it's a G here, it's a C there, and vice versa. So what nature has done, and this holds true for all plants, animals, fungi, is to protect these linear chromosomes. The blue strand wraps around like a paperclip and then tucks back in to mate with the red strand. It's kind of clever. And on an ultrastructural level, we see that there's actual binding proteins that mediate this, and there's other sheltering molecules. None of this is important. You just got to remember the paperclip. And last week we discussed why telomerase activation is not carcinogenic, and here we have absolute must principle here. So this bow tie with the squiggles, the double helix, is the chromosome. Here's the overhanging leading strand, that thing we just looked at, that's getting actively lengthened like an old Dymo label maker for your locker or your peachy notebook. This kind of gets longer and longer if it's a stem cell. This thing is a bunch of proteins, and it's a machine. So the question came up last week, why don't we just inject telomerase? Well, telomerase is a machine. It's assembled within that cell for use in that cell. It can't cross into cells, and it needs to be synthesized. Each one of these proteins needs to be synthesized, and it needs to be assembled into a little, like Lego, micro machine. So it's only for use within that cell and only for use within that cell's nucleus to repair that cell's DNA. The green is the RNA component, TERC, RNA component. That is the template, which is the printing press. It tells you which 6 base pairs are you going to print out in the case of humans. Maybe it would be 23 in the case of yeast. So this green thing is the RNA component, and this is the key to run the engine, which is the TERT, reverse transcriptase. The naming is somewhat troublesome, but RT basically is an engine. It's a mixture of a bunch of molecules. And RC is the RNA component, which is a single molecule, single RNA molecule. And this is the end that gets spliced. As we discussed last week, stem cells have a splice key, which works. It runs the keyhole, which changes the conformation so that the whole thing can start working. In a regular cell that's not immortal, the key is too long. It doesn't fit the keyhole, which is the case with that premature aging Discaron syndrome. And so the key is useless. And so in most of our cells, the key is useless. It doesn't matter if you have a lot of engine. You're not going to be able to write more DNA. Brief review of genetics. There was a guy named Gregor Mendel, a monk with some time on his hands, and he started breeding beans. And he found that, as humans have done for thousands of years in terms of husbandry of animals and plants, that if you cross certain plants, you get mixed traits. So if you have a green bean and a green bean, there's the green gene and maybe the not-so-green gene. So the children of this plant would be green, 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 because you only need one copy of the gene to be green, But if you had a not-so-green gene here and a not-so-green gene here, then 1 out of 4 times you'd get a white bean. Okay? And this holds true with people as well. So in a typical situation, let's say there are many diseases where the dad would be normal, 2 capital R's. The mom might be normal but with a hidden gene with a small r. So in this case, if you do the squares, you're basically talking about there's 2 chromosomes, one from mom, one from dad, chromosomes like this, and genes are just little sequences of DNA. So what Mendel was finding out was that you get 1 copy of a gene from mom, 1 copy from dad. So in this case, you can get 2 capital R's, totally healthy, 2 capital R's, double homozygous for the wild type or the healthy natural state. And you can also get a situation where half the kids are carriers because mom gives half her genes here, half her genes here. Kind of confusing, but it'll become useful shortly. This is a famous picture of Yalta just before the end of World War II, and it just shows some heavyweights here. Got Churchill, we got FDR, and Joe Stalin here. And so they kind of decide how they're going to carve up the world there. There's 3 people here. Blasco, the lead author, Grider, who worked with Blackburn, the Nobel Prize winner, and also DePino, who's now at Harvard Dana-Farber Institute, who published the article on the mice with telomerase activation. So these people, 1997, were all present at Cold Springs Harbor and learning about how to engineer mice. So what did they do? They took these mice and they knocked out their Turk. Remember, reverse transcriptase is the whole enzyme engine, but RC is just that green key. So they created mice that were deficient. Mom and dad gave them a deficient copy, whatever. They have no ability to make a good key. So they found out that these mice, as they went through generations, got older. The G1 was the first generation of knockouts. The second generation were born older, and with all the damage. The third generation were even worse, and the fourth generation were not even viable. So unlike the song from Bye Bye Birdie, what's the matter with kids these days, that's the thought you need to think about, because look at Larry King over here enjoying his wonderful children. And the point is that Larry King, his sperm, his germ cells are immortal, and they're darn good. There's good error correction. There's infinite telomerase activity, protected caps. So what they did was to take these mice and make them telomerase incompetent by removing the key. So every generation was worse. You know, we only have the perception as we get older that there's something wrong with kids, but in actuality they're just as good.
Why can't they be like we were, perfect in every way? What's the matter with kids today? Kids!
And in this case, the kids of the mice were progressively older and worse off. That's kind of a mean thing to do to mice, but it proved a point and built the next 14 years of research. So here is what that would look like. In this case, let's say the mom is a double knockout mouse, one of these genetically mutated G1 or second generation, no telomerase, getting older mice. You introduce a half-normal dad, and you mate them, and you get, again, knockout. Half the kids are knockout, still not able to grow their telomeres. Half now have recovered one gene from the half-healthy dad. These are restored, knock-in mice. So what they discovered, what Blasco discovered long ago, or maybe 4 years ago, was that if you took the bad mice and knocked it back in the third generation, well, look what happens. The normal telomeres are still long. The broken telomeres are still short. But the repaired genes, repaired. Knock in, long telomeres. And good news. Now we're switching gears to TERT. Again, it's confusing. Forget the TE part. Just look at the RT part, reverse transcriptase. That's the engine. So what Blasco did, and her resume just is a very interesting resume because she basically asks big questions, fundamental questions, and tests them. And her career alone, in my opinion, has advanced the understanding of this field more than any other single investigator. And she remains a close collaborator with Geron and with TA Sciences. And in fact, was featured last month in a news report picked up by the mainstream media regarding her test for critically short telomeres, which is commercially available in Europe. And the test was being touted as a test to tell you how long you're going to live, which is really not the case at all. But they did develop a novel test for measuring short telomeres way back in 2007, and now it's going to be available for people. So the reverse transcriptase enhanced mice, she took these mice that were enhanced in terms of their tumor suppression, P53, P16, P19. These are all the monitoring and the proofreading and the killing off of the bad genetically damaged cells. These are all very meaning that cells that probably wouldn't have had that were differentiated, she gave them that ability. And guess what? They lived longer and healthier, and they lived 40% longer. So it's the combination of having telomerase activity in previously telomerase inactive cells and the high level of error correction that made them all good. There was no increased cancer, and the lifespan was increased by 40%. Again, reverse transcriptase, we're not talking about the key anymore. We're talking about the engine. The Harvard mouse study that got a lot of press back in November and December of last year was by DePino, one of those guys that was president at Cold Springs. I think he came through, I want to say, Stanford, UCSF, and now Dana-Farber a year or two ago. So he's made a great study here of what happens when you take these engineered mice. These are not knockout Turk mice, okay, not like the ones they created in 97. This is knockout for TERT. Something about the reverse transcriptase is defective here in both copies from mom and dad. So they made these mice that age fast, but they included a lock and key mechanism, meaning they could turn it on at will. It was like a latent gene. So they created these mice with a lock, and the key was a tamoxifen derivative or a relative. Not important. It's just a steroid hormone that could activate the telomerase. And what they found, to their surprise, was that the mice got younger, like Benjamin Button. Not only did they not get older, which they were anticipating, just an arrest of aging, but they actually saw youthening, if we can coin that word. The skin, the pelts got darker. The neurological system, measured by smell and all these other things, got younger. And so this was exciting, and people said, well, that's neat. Wouldn't that be great if we could do that with people? Hello, we've been doing that with people since 2007. That's exactly what TA65 does. And that's why I believe, and my patients are believing, and the evidence is showing that people are getting younger. So how does TA65 work? Well, how do we know telomerase is active? There's something called TRAP, which is a telomerase reverse transcriptase activity protocol, I think. It's something like that. It just means you put it in a dish and you see if there's more activity. And the way you know that it's active is that new base pairs are added to the telomeres. This is a standard measurement that people do to measure activity. So if you want to know whether yoga or good exercise or sleep increases telomerase activity, you would use a TRAP assay. So the paper that came out in April by Blasco with the TA Sciences folks and Cal Harley, who's the lead scientist and discoverer of TA65 from Geron, showed that at 24 hours at this concentration, TA65 increased telomerase activity in these third-generation mice. So now we know, remember, that G3 are the ones that their parents were incompetent, but now being spliced with a half-normal dad, they have some telomerase activity. And if you could read this, I apologize for the size, it would show positive, negative. So third-generation mice, and this high bar showing that they have 2.5 times the telomerase activity. And that's good. Here's the double negative, G3, without the crossing with the normal, half-normal dad. There's just no activity there in the telomerase. And the interesting thing is that also this result did not persist at 5 days. So it's a transient effect. It translates to those of you taking TA65. It's not going to be there 5 days later. Okay, it's only for the 24 hours that you're taking it. And I think people who go on and off can attest to that. There is a certain immediacy. So what happened in 2011 April was she published these articles on the mice, but she did this just to dot the I's and cross the T's. She already knew what the result would be because she did this experiment in 2007 where she had the Turk knock-in mice, and she found that these histograms of the end-to-end fusions that we've talked about in the past, you went from this number to none. Okay, it's clearing these bad double chromosomes. End-to-end fusions to none. Fragments, breaks, still present, but these rings, they went to none. So something about making telomerase active by restoring the key allowed the cell to go through its final apoptosis, or killing off of those damaged chromosomes. So fast forward to 2011, the recent study, she found the same thing here, that signal-free number dropped when you added TA65. Critically short telomeres dropped, adding TA65. And this is something which is a signal of DNA damage, which would occur if you had, say, oxidative damage or just an uncapped telomere. There's a signal, and this shows that that signal was decreased greatly when you're taking TA65. Now, the good news is that although the effects of reverse transcriptase activity are only dependent upon the time that you're taking it, the repair is permanent. And so we know that because if you look here 2 years out, and this is 3 months after treatment, the control group still had a lot of short telomeres, had broken telomeres, although the treated group, even long after the stuff was out of their system, the TA65 group had less. So what's going on there? The only way to explain this is that, in my opinion, the damaged stem cells, the damaged queen bees, if you will, were kicked out and they were replaced by younger ones. And the copies of the younger cells, this is 3 months after treatment, they're making better copies of themselves. And that will come into play next week when we talk about weight, diet, and obesity, and how I lost 25 pounds, even, and it's impossible for me to put it back on. It's because the same cells are not present. They're less damaged now. They're healthier fat cells. So the point is that aging is an escalator that's going down, and when you take good care, you're able to stand still. If you don't take good care, the speed of the elevator catches up and you start to descend. But if you exercise, eat well, sleep well, reduce your stress, you run up the escalator, and then you are younger physically. It's kind of like pouring champagne into a tower of champagne glasses. And this is a big issue with people who maybe will notice an immediate effect, and then they'll say, well, I don't feel anything now. Well, you know what? You're trying to fill the glasses down here. You still have some damage that needs to get taken care of. And so you don't get old overnight. You don't get young overnight either. And that's the thing that a lot of people, dependent upon their psychic makeup, find difficult about taking TA65. They feel great initially. Maybe some arthritis goes away. Maybe they lose some weight, but they're like, I don't feel it. I just don't feel it. Some of the people with the most damage, I remember Chuck was one patient who had really horrible sleep. That sleep improvement didn't kick in until about 6 or 8 weeks because there was so much more in these champagne glasses that needed to be fixed. And so every night you only have so many things you can fix, and then you wake up and you go at it again, damage. And so it's a cumulative, gradual process of repair. But interestingly, although there are mechanisms to repair DNA, which we can get into someday, probably the main way TA65 is working is by allowing them to be destroyed, break the champagne glasses that aren't working, and and recruit ones that are going to be younger and healthier. And we've discussed that at length. It's called the mesenchymal stem cell recruitment, and that's in different podcasts, which I'll refer to in the archive. So that's it. Short one today. If anybody has questions, be happy to entertain those at this point. A question. Kent Meyer. It seems that TA65 is really better with HGH. I don't know. Does it seem that way? I know people who've gone off it after starting TA65, and I know people who remain on it. I think they're different, but perhaps complementary. Of course, HGH can be overdone. It can be supercharging, so I'm not a big fan of that. Ralph asks, with the replacements from the better stem cells, does the number of stem cells increase again? Think of it like this. You only have a certain number of queen bees per hive. So if there's a queen there and she is damaged, she'll often go into a state of, she can't copy because the DNA watchman, the P53, is keeping her from copying, but she's still doing her job. She's kind of phoning it in. She's kind of like a 63-year-old middle management person in state government. She just wants to phone it in. She's not really at 100%. So what happens is and they don't promote from within. The drones usually don't dedifferentiate, although that happens in muscle cells and satellite cells, as we discussed in podcast 11. What happens is there's a vacancy, and then these itinerant stem cells, these mesenchymal stem cells that are ready to go, they're college educated, they're smart, they can do this job, that, or the other. They come in and become the new queen bee in whatever niche that is. So anyway, it's a concept of stoichiometry. Somehow the cells signal each other to know that there's enough people here. We don't need anybody new, no new guys, and we already got a boss. Okay, and so what you need to do is kill off the bad cells in that niche. Usually there's only one less differentiated stem cell that replenishes all the drones around her with her bad or old DNA. So I hope that kind of answers your question. Okay, David from Manhattan Beach asks, what about menopause? Does the loss of estrogen create special problems for women? Yes, it does. Unlike andropause, which is a made-up thing, not recognized by the World Health Organization, women about the age of 50 run into a wall called menopause, and their ovaries really stop functioning to produce hormones. They still have hormones from the adrenal gland, which can be converted into all the different steroid hormones, such as testosterone, estrogen. But it drops pretty much precipitously. The upshot of which is decreased bone density, decreased energy. Sometimes the mood, especially during the transition, is poor. Hair quality, skin, nail, hair, especially in the sensitive tissues below the waist, it can lead to incontinence and dryness, etc. So Kent is on, but we only hear his radio. Okay. Question from Kenneth. In the TA65 supplement, do you continue to take it for life, or is there a specific time frame that you take it and then stop? Excellent question. Well, as we discussed here, the repair is permanent. So these mice were treated and weren't taking it anymore, so the trap assay was no different. They weren't getting any benefit. And yet, you measure their telomeres, they continue to have fewer short, critically damaged telomeres. So the question is, do you continue to take it? Well, I would, and I do plan to, because as long as you're still using the car, driving it, taking it out, it's still getting old. So why not reverse or, stop that aging process? It's a matter of finances, but, 20 years from now, I'm sure Monsanto will have engineered corn to produce high yields of this substance, and it'll just be a political issue whether people will be entitled to stay young. I mean, it raises huge social questions. Although, in reality, the Medicare problem would be solved, but you'd have a lot of unemployed doctors and insurance salesmen. So, anyway, this is way above my pay grade, but certainly, whether they're deserving or not, most people could benefit from a healthier lifestyle while they're here, and it definitely was something that I would consider taking indefinitely. So, rust never sleeps, as Neil Young once said. So you're always rusting, you're always oxidizing, and you're always aging. So why not continue taking it if it's money's not an issue? Don Wright, if I cut myself shaving, will I heal with a small young spot? I don't know what you're asking. When you cut yourself shaving, wounds tend to heal much faster, but that's because the cells are rapidly producing and they're healing more efficiently, and that we've noticed quite a bit. People who get cuts and things like that heal incredibly fast, and that would extend to ligaments, bones, soft tissue injury. So were there any questions about the slides? I know they're highly technical. I think everyone got the paperclip thing, which is really cool. And, I wish we could go into more of Blasko's research because, this seems like it's pretty down to the nitty-gritty, but all the stuff that people know about in terms of enzymes and genes, they look at the black box and they see what comes out. But the level of understanding that Blasko has contributed is astounding. And she really asked a simple question, does the experiment? And so there are actual players that we know have specific roles in the winding and unwinding, the copying, the DNA damage. And one of those interesting is the BRCA2 gene, which has a direct role in capping the telomere. So the reason why that breast cancer gene is so critical is because it's involved with the capping and the uncapping. And here is the slide that everyone should understand intuitively. Larry King's sperm, perfectly long telomeres, and his kids' telomeres, etc. Premature aging kids, rapidly decreasing telomeres. G234 mice, rapidly decreasing. Regular cells, somewhere in the middle. Not immortal, not rapidly aging, but really not going to live forever. They're the Hayflick limit, but probably not as a result of their own telomerase activity, but the fact that the cells are usually derived from a stem cell, which she herself is aging. So she's not a straight line across like the perfect copies of the sperm and eggs. She's kind of on a downward slope too. Question from the chat. Do cells have to regenerate to increase telomere length? No, there's certain cells that are just doing that all the time, like your red blood cells, your skin cells, your intestinal cells. They have some telomerase activity, even though they're not what typically we consider stem cells, like 1 in a million queen bees. So no, it's an ongoing process of increasing telomere length if the engine is running. Ken Shepard asks, how critical is it to avoid telomerase inhibitors like resveratrol and others while taking TH65? I don't think it's critical at all. TH65 is a very benign, very harmless small molecule that has a limited scope of action. Resveratrol, in my opinion, is green cheese from the moon, basically. I mean, it's just this ridiculous thing that, pretty sure the moon's not made of cheese, but apparently resveratrol got way ahead of itself in terms of somebody saying, hey, French people drink wine, they don't get as much heart attacks, therefore the wine must have something that, it's ridiculous. From an epidemiological standpoint, ridiculous. From a scientific standpoint, ridiculous. So the studies, they poured tens of millions of dollars into studying why, and I don't know why they even picked resveratrol, but all these great people assumed that it was true and went out for a decade to prove that it was true, and it turns out that none of it is true, and it's just a poison, and the reason why your cells may live longer is because they're poisoned and they go into crisis, which is one of the very popular ways to live longer is to starve yourself, caloric restriction. But yeah, I mean, if you're in a hunker down, low metabolism, hibernation mode, your cells and you are going to live longer, but it's not as good a quality of life. But a large resveratrol study was abandoned, so resveratrol, in my opinion, is not good. It was a cure in search of a disease, and there's no mechanism whereby it would be good. And for people to say that it's a telomerase inactivator, this is ridiculous. It has no role whatsoever because you could put anything you want into a trap assay. You could put Tabasco sauce or Tylenol. Yeah, and maybe there'll be a downstream effect, but that doesn't mean it's directly related to either TERT or TERK. So I would say you want to take supplements, that's fine. I don't think they're helpful, but I wouldn't say that something specifically is a telomerase inhibitor or a telomerase activator unless there's really good reason to think it's got a direct role in that. Oh, and if you think that was bombastic, wait for next week when I talk about diet and obesity, because basically the whole thing will be debunking what people intuitively take to be true about eating. It turns out that actually, I think in podcast 6 we talked about the fact that the food pyramid was basically agribusiness's attempt to BS us. And since podcast 5, I'm happy to say that the Big Brother, U.S. government, has rescinded the food pyramid and gone back to a paleo diet recommendation, which since Dr. Atkins in the 70s, we've known that it's all about the glycemic index. It's not about fat and carbs and all that. Really, we're not what we eat. And next week you'll find out that a lot of things that people think they know about nutrition are not true. And the simple proof of the fact that we're not what we eat is that we would all be cannibals and vegetarians would all be vegetables. So you basically, your bodies are metabolic machines and they create fat, carbs, proteins from eating fat, carbs, and proteins. And that's what it's all about. But next week we'll talk to a bunch of people who lost weight or even gained weight and discuss what the real deal is with nutrition and with obesity. Any other questions? So yeah, food pyramid in the toilet. All right, so we got everyone on board now. All these wonderful. Actually, thanks for everyone that tuned in. There's a lot of names here I see, and I really appreciate you guys tuning in. It's hard to talk to an empty room. Any questions? Bob, you're always having audio problems. What's up with that? Maybe I need to send you some batteries for the hearing aid. Just kidding. So here we go. Everyone remember the genetics, the Yalta, Larry King, what's the matter with kids these days? Mice that were broken and then fixed. Super mice with extra reverse transcriptase. Harvard super mice getting young, reverse transcriptase restored. Telomerase is activated transiently while you're taking TH65 in mice. And the way it's working is by reducing the proportion of signal-free, damaged, short, these donuts, these double ends. It's killing off some bad cells, I think. And that repair is permanent. So, anyway, stick with the TH65 if you're taking it. Really, I feel like it's important to have a doctor to talk to, and if you buy it from elsewhere, I think you'll have a hard time getting a doctor on the other side of the line, whereas, we are pretty much always talking and emailing and listening with our patients, and so we can share the information better. It's better to have a doctor helping you than to just mail order and and flush it down the toilet if you're not even absorbing it. So we're happy to take any Bioness patients, and it's complimentary as long as you're ordering with us. And I just think if you're going to spend that kind of money and really take that kind of powerful supplement, you want to have somebody following it. You just don't want to be putting that in your body and having no effect, or maybe an unpredictable effect, or an effect that you don't know, is this normal? So it's a big deal. It's not something that I would just do it yourself and just mail order, and because there's a lot going on here on different levels, and it helps to have somebody with a little experience who's going to listen. Kenneth, thank you for listening.